dsR, also known as endoribonuclease dsR or helicase with ribonuclease motif, is an enzyme that in humans is encoded by the dsR1 gene. Being part of the ribonuclease III family, dsR cleaves double-stranded RNA, dsRNA, and pre-microRNA, pre-miRNA, into short double-stranded RNA fragments called small interfering RNA and microRNA, respectively. These fragments are approximately 20 to 25 base pairs long with a 2-base overhang on the 5 ends. dsR facilitates the activation of the RNA-induced silencing complex, RISC, which is essential for RNA interference. RISC has a catalytic component Argonaute, which is an endonuclease capable of degrading messenger RNA, mRNA. Discovery. dsR was given its name in 2001 by Emily Bernstein, a graduate student in Gregory Hannon's lab at Cold Spring Harbor Laboratory, who sought to discover the enzyme responsible for generating small RNA fragments from double-stranded RNA. dsR's ability to generate 22 nucleotide RNA fragments was discovered by separating it from the RISC enzyme complex after initiating the RNA pathway with dsRNA transfection. This experiment showed that RISC was not responsible for generating the observable small nucleotide fragments. Subsequent experiments testing ribonuclease III family enzymes' abilities to create RNA fragments narrowed the search to Drosophila CG 4792, now named dsR. dsR orthologs are present in many other organisms. In the Mospis comatrella patens DCL1b, one of 4 dsR proteins, is not involved in mRNA biogenesis but in dicing mRNA target transcripts. Thus, a novel mechanism for regulation of gene expression, the epigenetic silencing of genes by mRNAs, was discovered. In terms of crystal structure, the first dsR to be explored was that from the protozoan Giardia intestinalis. A POS domain and 2 ribonuclease III domains were discovered by X-ray crystallography. The protein size is 82 kDa, while it is larger in other organisms, for example, it is 219 kDa in humans. The difference in size from humans to G. intestinalis dsR is due to at least 5 different domains being present within human dsR. These domains are important in dsR activity regulation, dsRNA processing, and RNA interference protein factor functioning. Functional domains. Human dsR, also known as HS dsR or dsR1, is classified a ribonuclease III because it contains both helicase and POS, Peewee/Argonaute/Sweel, domains. In addition to these domains, HS dsR contains 4 other functional domains, 2 RNA domains and 2 double-stranded RNA binding domains, DUF283 and dsRBD. Current research suggests the POS domain is capable of binding the 2 nucleotide 3 overhang of dsRNA while the RNA catalytic domains form a pseudodimer around the dsRNA to initiate cleavage of the strands. This results in a functional shortening of the dsRNA strand. The distance between the POS and RNA domains is determined by the angle of the connector helix and influences the length of the microRNA product. The dsRBD domain binds the dsRNA, although the specific binding site of the domain has not been defined. It is possible that this domain works as part of a complex with other regulator proteins, TRBP in humans, R2D2, LOX in Drosophila, in order to effectively position the RNA domains and thus control the specificity of the dsRNA products. The helicase domain has been implicated in processing long substrates. Role in RNA interference. MicroRNA. RNA interference is a process where the breakdown of RNA molecules into mRNA inhibits gene expression of specific host mRNA sequences. mRNA is produced within the cell starting from primary mRNA, pre-miRNA, in the nucleus. These long sequences are cleaved into smaller precursor mRNA, pre-miRNA, which are usually 70 nucleotides with a hairpin structure. Pre-miRNA are identified by DGCR8 and cleaved by DROSHA to form the pre-miRNA, a process that occurs in the nucleus. These pre-miRNA are then exported to the cytoplasm, where they are cleaved by dsR to form mature miRNA. Small interfering RNA. Small interfering RNA, sRNA, are produced and function in a similar manner to miRNA by cleaving double-stranded RNA with dsR into smaller fragments, 21 to 23 nucleotides in length. Both mRNAs and sRNAs activate the RNA-induced silencing complex, RISC, which finds the complementary target mRNA sequence and cleaves the RNA using ribonuclease. This in turn silences the particular gene by RNA interference. cRNAs and mRNAs differ in the fact that cRNAs are typically specific to the mRNA sequence while mRNAs aren't completely complementary to the mRNA sequence. mRNAs can interact with targets that have similar sequences, which inhibits translation of different genes. In general, RNA interference is an essential part of normal processes within organisms such as humans, and it is an area being researched as a diagnostic and therapeutic tool for cancer targets. Disease. Macular degeneration. Age-related macular degeneration is a prominent cause of blindness in developed countries. DICER's role in this disease became apparent after it was discovered that affected patients showed decreased levels of DICER in their retinal pigment epithelium, RPE. Mice with DICER knocked out, lacking DICER only in their RPE, exhibited similar symptoms. However, other mice lacking important RNA pathway proteins like DRASA and PASHA did not have symptoms of macular degeneration as DICER knockout mice. This observation suggested a DICER-specific role in retinal health that was independent of the RNA pathway and thus not a function of C/mRNA generation. A form of RNA called LuRNA, the RNA transcripts of LU elements, was found to be elevated in patients with insufficient DICER levels. These non-coding strands of RNA can loop forming tRNA structures that would be degraded by DICER in a healthy retina. However, with insufficient DICER levels, the accumulation of LuRNA leads to the degeneration of RPE as a result of inflammation. Cancer. Altered mRNA expression profiles in malignant cancers suggest a pivotal role of mRNA and thus DICER in cancer development and prognosis. mRNAs can function as tumor suppressors and therefore their altered expression may result in tumorigenesis. In analysis of lung and ovarian cancer, poor prognosis and decreased patient survival times correlate with decreased DICER and DRASA expression. Decreased DICER mRNA levels correlate with advanced tumor stage. However, high DICER expression in other cancers, like prostate and esophageal, has been shown to correlate with poor patient prognosis. This discrepancy between cancer types suggests unique RNA regulatory processes involving DICER differ amongst different tumor types. DICER is also involved in DNA repair. DNA damage increases in mammalian cells with decreased DICER expression as a result of decreased efficiency of DNA damage repair and other mechanisms. For example, cRNA from double-strand breaks, produced by DICER, may act as guides for protein complexes involved in the double-strand break repair mechanisms and can also direct chromatin modifications. Additionally, mRNA's expression patterns change as a result of DNA damage caused by ionizing or ultraviolet radiation. RNA mechanisms are responsible for transposon silencing and in their absence, as when DICER is knocked out/down, can lead to activated transposons that cause DNA damage. Accumulation of DNA damage may result in cells with oncogenic mutations and thus the development of a tumor. Other conditions. Multinodular goiter with schwannomatosis has been shown to be an autosomal dominant condition associated with mutations in this gene. Viral pathogenesis. Infection by RNA viruses can trigger the RNA cascade. It is likely DICER is involved in viral immunity as viruses that infect both plant and animal cells contain proteins designed to inhibit the RNA response. In humans, the viruses HIV-1, influenza, and vaccinia encode such RNA-suppressing proteins. Inhibition of DICER is beneficial to the virus as DICER is able to cleave viral RNA and load the product onto RISC, resulting in targeted degradation of viral mRNA, thus fighting the infection. Another potential mechanism for viral pathogenesis is the blockade of DICER as a way to inhibit cellular mRNA pathways. In insects. Insects can use DICER as a potent antiviral. This finding is especially significant given that mosquitoes are responsible for the transmission of many viral diseases including the potentially deadly arboviruses, West Nile virus, dengue fever and yellow fever. While mosquitoes, more specifically the Aedes aegypti species, serve as the vectors for these viruses, they are not the intended host of the virus. Transmission occurs as a result of the female mosquito's need for vertebrate blood to develop her eggs. The RNA pathway in insects is very similar to that of other animals. DICER2 cleaves viral RNA and loads it onto the RISC complex where one strand serves as a template for the production of RNA products and the other is degraded. Insects with mutations leading to non-functional components of the RNA pathway show increased viral loads for viruses they carry or increased susceptibility to viruses for which they are hosts. Similarly to humans, insect viruses have evolved mechanisms to avoid the RNA pathway. As an example, Drosophila C virus encodes for protein 1A, which binds to dsRNA, thus protecting it from dicer cleavage as well as RISC loading. Heliothese viruscens ascavirus 3A encodes an ribonuclease 3 enzyme similar to the ribonuclease 3 domains of dicer, which may compete for dsRNA substrate as well as degrade dsRNA duplexes to prevent RISC loading. Diagnostic and therapeutic applications. Dicer can be used to identify whether tumors are present within the body based on the expression level of the enzyme. A study showed that many patients that had cancer had decreased expression levels of dicer. The same study showed that lower dicer expression correlated with lower patient survival length. Along with being a diagnostic tool, dicer can be used for treating patients by injecting foreign dsRNA intravenously to cause gene silencing. The dsRNA was shown to be delivered in two ways in mammalian species such as mice. One way would be to directly inject into the system, which would not require dicer function. Another way would be to introduce it by plasmids that encode for short hairpin RNA, which are cleaved by dicer into dsRNA. One of the advantages of using dicer to produce dsRNA therapeutically would be the specificity and diversity of targets it can affect compared to what is currently being used, such as antibodies or small molecular inhibitors. In general, small molecular inhibitors are difficult in terms of specificity along with unendurable side effects. Antibodies are as specific as dsRNA, but it is limited by only being able to be used against ligands or surface receptors. On the other hand, low efficiency of intracellular uptake is the main obstacle of injection of dsRNA. Injected cRNA has poor stability in blood and causes stimulations of non-specific immunity. Also, producing mRNA therapeutically lacks in specificity because only 6 to 8 nucleotide base pairing is required for mRNA to attach to mRNA. Dicer-like proteins. Plant genomes encode for dicer-like proteins with similar functions and protein domains as animal and insect dicer. For example, in the model organism Arabidopsis thaliana, 4 dicer-like proteins are made and are designated DCL1 to DCL4. DCL1 is involved with mRNA generation and dsRNA production from inverted repeats. DCL2 creates dsRNA from cis-acting antisense transcripts which aid in viral immunity and defense. DCL3 generates dsRNA which aids in chromatin modification, and DCL4 is involved in transacting dsRNA metabolism and transcript silencing at the post-transcriptional level. Additionally, DCL1 and 3 are important for Arabidopsis flowering. In Arabidopsis, DCL knockout does not cause severe developmental problems. Rice and grapes also produce DCLs, as the dicer mechanism is a common defense strategy of many organisms. Rice has evolved other functions for the 5 DCLs it produces, and they play a more important role in function and development than in Arabidopsis. Additionally, expression patterns differ among the different plant cell types of rice, while expression in Arabidopsis is more homogeneous. Rice DCL expression can be affected by biological stress conditions including drought, salinity, and cold. Thus, these stressors may decrease a plant's viral resistance. Unlike Arabidopsis, loss of function of DCL proteins causes developmental defects in rice. L proteins causes developmental defects in rice. L proteins